Okay, so today is the last lecture for Tuesday. We are going to talk about networks. Next week, next Tuesday, I will give you time for yourself, and then on Thursday you are going to do the presentation. So Thursday will be next Thursday, 27. Yes, April 27. We will be in the room 115, so we'll be this presentation. So we will have a score sheet. So this will have several categories, significance. So that's your topic, how significant it is, and also the topic innovation. Okay, and approach. So the methodology you are going to take to answer the hypothesis you are going to test. The data you can get from anywhere, and this is single cell data and related. You try to reflect what you learned from the class. And the last category of this criteria is the performance. How are you going to deliver your message clearly and attractively to the audience? From the last several semesters, I found this very interesting and intriguing event, and students enjoyed that a lot. And you will have a score sheet for every one of you, and you score for others, and you rank who's the top in terms of the significance, who's the second in the significant, like who's the top in the innovation aspect, who's the second, something like that. You give the 1, 2, 3 rank, and I and the Yunjian will go back and depends on the rank and to calculate overall score and for everyone. Okay. So that's a presentation, and the Tuesday I will also send you some link. One is about so Tuesday over here. Okay. 25, no class, but the YouTube links. Okay. One is the regression. Okay. Regression, the last one. So you will do this. I watched the video I took, and also the homework for the classifier. Remember, I gave you a big spreadsheet for classifier homework. So in that video, I will explain how you should do and what should you achieve through the practice. Okay, so week 13 network. Yes.
Is it necessary for us to download the data from the article on labeling?
Yeah, you can do that.
Like if I can use only read the things, the data set of the materials.
Yeah, but you have to have your own hypothesis, what you want to achieve. So whether you achieve your goal or not, that's up to you. It depends on the quality of data, and you just show people you're thinking towards that direction, and it's a unique problem. And if we don't have that in eukaryotes, maybe you want to find eukaryotes which under the infection of bacteria, and you say, okay, I also work from eukaryotic cells, and before and after infection, what I see. So you can do both. That will make that very interesting. Because we work from two sides, pathogen and host response.
Okay.
Yeah. But to be creative, so there's no yes or no answer, but the audience will decide, yourself will decide who's the top in terms of innovation, approach, performance. Yeah. Okay, so we are going to go through a lot of this network-related stuff. I don't know how many of you already have this network analysis experience. We will define and mention a lot of centrality measurements that for to calculate, to quantify the value of each node in the network. Okay, depends on some topological properties of these nodes. We will mention WGCNA. This is a very popular R package, has been widely used since 2005. So it has been at least 2 decades of these applications. So many genetics research for networks have been using this package. And you probably will recognize some of the figures generated by this package. So it's not the best one, and it was a very popular one. So I have to mention it. And so you know what's behind this approach, how the data get smoothed out and to so-called make the noise, less noise and more signal. We will mention the community detection. That's to detect sub-modules among a big network. So after that, we'll mention the advantage of single-cell gene regulatory network compared to traditional co-expression gene regulatory co-expression network created using traditional RNA example. We will mention the Gene3 and its principle behind this is using the tree to do this classification and the regression. Then we have multi-layer networks, and then at the end we will re-illustrate what we have done using network for single-cell data. This is a single-cell 10-fold suite package. There are several applications. You probably already heard about this interaction detection. Yongjian presented last time here, and that's one of the applications for detections, cell-cell communications using network information. Okay, so in order to build this biological network, we collect signals. So in this case, the example is from this functional MRI time series data, and you collect the signal across the time at a different position in the brain, and you get this time series data, one from the location red and another from blue. Once you have these two curves, you can start to build the correlation. So these two signals always move up and down together, then they're strongly correlated. Then you can put the position, for example, this is strongly correlated, then you put the correlation in the middle of this. You can sample a lot of places. So once you have enough samples, you can create. So if, for example, there's n samples, there's only 2 points that have been shown. But if you have n samples here, you build a n by n pairwise correlation matrix. So from this matrix, you do a filter. So you start to get rid of some of those weak links. Correlation is weak, so you can set up a cutoff position, and then you throw away a lot of weak links, and then create a so-called adjacency matrix. This is a representation for the network. So once you have adjacency matrix, that's a network, you can do downstream analysis to study the function of the network. For example, in terms of this example, some of those modules maybe there are several locations, they are linked. Okay, so this is one of the this open question. Once you have a network, what you can do with this? In our data, every point is a gene. So you start to look at the co-regulated module of the genes. In other networks, there may be a social network, and then there's a group of people, and the company, the more there are different ways to see, okay, what kind of function you want to get from the network. So this is just an overview. On your right, this adjacency matrix, big A is matrix, and this matrix is a binary. Once you have one, then you know the node 1 and the 2 is connected because this position is 1, 2. Okay, and also 2, 1, 1 is 2 to 1 or 1 to 2 are the same. So this network is undirected. The adjacency matrix is symmetric. And the 2 and the 3 is linked. So you found the 2 and the 3 is 1. 3, 2 is 1. So in this way, you describe topology of this network using a matrix. Okay, so those nodes and those links are the terminology you see in the network analysis. When you work on the graph, they're same thing, okay? And then you see, okay, there's a vortex in the edges. But sometimes people just miss all this terminology. It doesn't matter. So overall, so network analysis is, to me, is relatively simple for biologists to understand because they are straightforward, intuitively simple. You know, you want to describe the link, you put one. Compared to PCA or other SVD, eigenvalue decomposition, etc., it's more difficult to biologists. So network in general, you may see a lot of formulated in mathematical way, but once you know what this present for, you should be able to get the idea very easily. So this graph is symmetric. So this adjacent matrix is symmetric and also undirected. But you can have a directed network, and then whether you have a link from 2 to 3, without a link from 3 to 2, then you make it asymmetric. You can also weight those values. Instead of 1, you can say, okay, this is a strong link, I put 1.5. And this is even stronger link, I can put 3.5, something like that. Okay, you can have a weighted network. And also you can have a signed network. Okay, rather than all the positive weight for every link, you can have negative. So if you have suppression relationship, say expression of 5 will suppress the expression of 4. So this link may have a -1 or -1.5 weight. Okay. So depends on what type of this network you have. Directed, weighted, signed network. So the analysis method will slightly different from symmetric, undirected, unsigned network. Okay, some of these details of the annotation of the computation will change a little bit. So now we assume you already build a network. there are many, many different ways to build a network. So once you have done, for example, you create a network. So what kind of question you can ask? So we will compute the values for every node. So those nodes will reflect the value of those vertices, okay, the nodes in the network. For example, the simplest centrality value you can compute is called a degree. This purple node have 4 links around it. So the degree of this node is 4 compared to this node here, the degree only 1. So if you believe the fully connected mode connection means more functional importance, then this purple node will be more important compared to this node. This is only 1 degree. But this is not the best way. And sometimes you want to say, okay, I want to compute the so-called betweenness centrality. Well, the degree for this blue node is not as high as red and purple, but it's in the critical position because you have a lot of links. If you go from this position to here, you have to go through the blue node. So we need a way to compute the so-called betweenness centrality. So you are in the critical position in the graph, and a lot of the paths, shortest paths from one node to another have to go through you. So this is another way to compute a centrality, okay? So for each node, you can describe it. For example, you use degree and also some betweenness centrality or closeness centrality or clustering coefficient. For each node, you can have a lot of different parameters, different features to describe the value of each node. Okay. So let's see, I'm going to use examples, simple examples to describe a few centrality computations so you get some idea how these values have been computed. As I said, relatively simple because the concept can be visualized. You have a graph, you see that. So as long as you know each of this parameter, what they stand for, and you should be able to get the idea. So now we are going to compute the closeness centrality for one of the nodes in the network. So this node X, for example, we are going to compute closeness centrality for A, okay? So you need some values. So this value is the distance from A. So now we replace X to A, okay? So Y is all the other nodes, not A in the graph. So now the Y for this example stands for B, C, D, E, okay? Now you have to get the distance from A to B, A to C, A to D, A to E. So, and then sum them up and then put 1 over it. So how can you compute this centrality value for closeness centrality for A? So you have a step from A to B is 1, right? A to C is 2. A to D is 3, A to E is 4. So you put all the distance from the node you want to compute its centrality to all the other nodes, and then sum them up, and then use 1 to divide by it for it. Then you got the centrality value for A is 0.1. So you do the same for B, and then you do the same for C. Every time you just replace Y to the other nodes around it. Okay. After computation, you will find, okay, A and E have the same value, 0.1, while C have the highest value, will B and B have a greater than 1, A and E, but smaller than C. So this value indicate whether this node is in the center of your network. Okay, if you are in the center, you distance to all the other nodes on average will be small, and 1 over this small value should be a larger value. Well, if you on the edge or the boundary of this network, the distance from you to all the other nodes are relatively far, and the large distance translate to a small value. Is that easy? Okay. It's widely used centrality, but it's very easy to compute, right? Another, this is called harmonic centrality. Basically, you are going to compute a harmonic mean for the data, okay? Almost the same as this one. This one, you just get the total distance and 1 over it. But this one is you have 1 over the first distance, 1 over the second distance, that you sum up all this fraction, okay? So this is the computation for the harmonic centrality, and then you get 2.5, 2.5, 3.5 those. And if you have this isolated, disconnected subnetwork, you have 1, 1 in there. So next, I'm going to show you an example to illustrate what the powers for the adjacency matrix. I don't think I'm going to use Blackboard. Let's just use this on-screen illustration. For example, you have a network on your left here, okay? This is a directed network. The reason is you have a link from a 2 to 1, but not 1 to 2. Okay, 2 to 1, so you have 1 here. That means from a position 2 here, and you have a link to 1. You don't have the other way around. So on and so forth. So you have your adjacency matrix M. Okay, so now I'm going to multiplicate M by cell. Okay, so this is a squared M. So you know how to do the matrix multiplication. You put the squared matrix here, squared matrix here, so you still have this squared matrix. If M is n by n, you multiplicate by another n by n. You can do it, and the product is still n by n, right? Let's see. So if you do this, you get this. Okay. So what's the number in there? What does that mean here? There is explanation for why what the 2 means here. Okay. Now let's see. Okay, there's 2 here. So that's a node 2. There's node 2 to 1. So so the value here means it's a parse from a position 2, I think 2 from 1 to 2. Okay. I remember this. So this position is from a 2 to 1. And this should be 1 to 2, okay? In your adjacency matrix, there's no path you can go from 1 to 2 because they are directed. You cannot go backward. But once you compute multiplicate it itself, so you squared it and then this become 2. What that means is you have 2 different ways to go from 1 to 2 by 2 steps. Okay, this 2 here means 2 steps. Let's see from 1 to 2, why you have 2 different ways to go. So you can go from 1 to 3, and then from 3 to 2. So this is one way, 2 steps. You can also go from 1 to 4 and from 4 to 2, 2 steps. So the number here to show you how many different ways you can go from node 1 to 2 by 2 steps. Now you can validate, say, okay, this position, this is from 4 to 1. From 4 to 1, you should have by 2 steps, you should have 3 different ways. From 4 to 1, by 2 steps, so you can go 4 to 2 to 1, one way. 4 to 5 to 1, another way. 4 to 6 to 1, 3 way. So the value there is you have 3 different ways to go from one node to another by 2 steps. So you can do multiplicate by another n. So you can do this 3, okay? You also get n by n matrix. So that matrix will tell you from one node to another how many different ways you can go through 3 steps. Do you think that information is useful? So the adjacency matrix is one step. Tell you the direct link from one node to another. But once you take multiplicate by itself, you get the value which is 2 steps. So you start to review the indirect link across the network from one node to another. And also you know how many different ways you can go. So if you do have this connectivity from signal, you have one signal you can separate into, for example, you have 3 different ways to go one node to another by 2 steps. You probably can separate this signal into one third each, and then go to this another node. So it start to reflect indirect link by multiple steps across network. So we will do adjacency matrix, which is step 1, and the squared, step 2, cubic, steps 3 steps, and you can force power, and the 4 steps. You can keep doing that until you reach infinity. Yeah.
Sure. What gives the direction for the arrows?
Oh, depends on which you can consider this is the source, that's target, or other way around. So if you think this is a source, so that's 1, 2, 3, 4, 5, 6. So that's 6. And this 6, and that's 1. So 6 to 1. So that's a direction. And from 1 to 6, we don't have this. Yeah. Is that clear? So you can define yourself, say I will consider the position in the rows as the source, and then the coordinates for the columns as targets, the position from 6 to 1, this one here. But if you transpose it, then you just define a slightly different way. Yeah. Okay, so this spectrum measurement called the Kasu index is using these properties I just described. So this Kasu index, for example, you want to compute the Kasu index for one of the nodes X in the network. You will consider adjacent matrix A here, then you take power from 0 until infinity. Okay. So 0 will be identity matrix, 1 will be adjacent matrix itself. Then you take 2 power 2 and 3 until the infinity. And then you sum it up. Basically, you have a lot of this matrix, and then you sum it up. And the link between one node to another, the values, will reflect either direct link, the power, and also all the possible links from one node to another by multiple steps. By step 2, step 3, all the possible links will be summed up. So this gave you a total number of works by different numbers of paths. You can go from this node to another by taking 50 steps, and you know it's possible by adding them up. So you reflect the total possible links from one node to another by 1 step, by 2 steps, by 3. At the end, you get the total. And then you compute, this is called attenuate. So you put a slightly weight value. If you have a longer steps, you make this smaller. And the direct link, the weight is higher. So you sum it up. Okay. So this reflects. So you do this for X to all the nodes, other nodes, and take us together. So this value will reflect the value of the nodes X you are trying to compute. This is called Kazu index to show you the connectivity overall from one node to all others. Sum together. Yeah.
Is there a significance of the diagonal?
This one here.
Is the diagonal that we get 2, 4, 3, 1, 2, or the 2, 4, 4, 0, 0?
Diagonal. 2, 4. Which one?
Both for A square and A2. The diagonal that we get. Like,
And where's all zero? Usually for the.
like for A1, those are all zeros. Because,
Oh, you mean this position here? Zeros in the diagonal.
yeah.
Okay, so
For A square and A2.
so this is a self link. So if you have a loop and link to yourself, you'll put a non-zero there. But when we talk about there's no self link, then the adjacent matrix, the diagonal position always zeros.
But in A square and A2, there are diagonal.
So once you have this, is that true? Let's see whether. So that means you come back to yourself using 2 steps. I don't know whether that makes any sense. Yeah, I have to compute whether this is correct because yeah, so for this network,
Usually that it's the beginning. In this case, I think it is the beginning for A square. One is connected to 2 links.
oh, you say this value here is the degree of each node that they put in there. It's not the power of this adjacent matrix itself.
I think so.
Yes, so let's check whether this is true. Looks like they put the degree for each node in the diagonal, but that's also changed. Let's try this, Thursday, in MATLAB and see whether. I don't think when you do the power for this, you get any values in the diagonal position.
Yeah, but they have both values. That's why I was wondering.
Interesting. That's a good observation. Maybe, yeah, let's check this. Okay, so once you have a network, you can compute those different types of centrality over the node. For example, you have this degree node values and put in this node, and you know, okay, this position here, a lot of high degree nodes cluster together. Well, when you use different like closeness, and you found the highest value center is in somewhere else. And then if you use others, for example, this is the harmonic centrality, this is the causal index, you get all different kind of values. Each node you can have select different centrality to describe it. And some of this correlated, for example, degree, oftentimes positive correlated with values in between this centrality. Most of the time you have a high degree, you have a high closeness centrality, for example. But there are also subtle difference. Each node in the network may be described with multiple centrality values and make them unique features. Okay. And also there are many new centrality measurement has been invented every day. If you look at those journals, maybe not every day, it's a little bit, it's not really, but I would say every month you should have found some centrality, a new centrality definitions in the paper of the network. For example, this is called leverage centrality. It's the difference between the degree of given I and J and the difference and there's and anyway, so this you can invent your own centrality if you like, as long as you say, okay, my definition of this particular way to compute the centrality will reflect one of the aspects in the topology position in the network. You can justify it, use some examples, then you're okay. All right. Okay, so as I said, this WGCNA is called Weighted Gene Co-expression Network Analysis. This package contains several steps, and this has been around since 2008. It's our package, and I don't know whether you have ever seen some like this. Who have ever seen some figures? Looks like this. No, not yet. But for the genetic study, then when you have multiple samples, you want to do, okay, I have a finished differential expression analysis. What we can do normally, a geneticist just put this expression matrix into WGCNA to compute the co-expression between genes. Then they come up with this network, and then once you have network, you can do this clustering. You put genes which closely or strongly connected together as a module, and then you start to annotate. So these genes in the same module, whether they in the gene function, the enrichment analysis, they share in the same pathway or same signaling subnetworks. Okay. Then we start to do the relationship of sometimes they also have this sample information getting involved. Okay. So the first step is to construct gene regulatory network. As I said, the traditional gene analysis using bulk RNA-seq, you never get enough samples. So a lot of those correlation you constructed I believe many of them should be false positive. For instance, if you want to have some genes correlation between, if you have only two conditions, control and the case, and you see, okay, those green genes, 3 green genes was highly expressed in control and they downregulated at the same time in case. And then you just build these links between the 3 genes, okay, because they call up or down regulation. And the 3 red genes as well, okay, they regulated to the same direction. And then you just make a link between them. Because you never get enough samples, so this signal is really low power, and you can put 3 dots easily, and oftentimes by random chance you get some correlation easily. So I never trust any of those WGCNA results. Okay, it's one of the way to analyze data, but don't believe it. So you constructed, see, okay, those purple genes, they all same time, they all co-regulate up and down at the same time. But because you only have 2 conditions, it's very easy to get a gene up and down. It's very different if you have 1,000 different categories, and those categories, for example, you have information from a time 0, okay, control, time 1, time 2, time 3, you have 1,000, then those correlation should tell you something. But in this case, you have a case control, all right? So don't trust too much about those links, but that's just one way you can analyze. So you start to build this network, and then within this big network, you start to recognize those modules in there. Okay. So that's the steps you want to take. So the network is constructed by correlation, and then you have a cutoff because you compute a correlation between genes. Every pair of genes, you can have a correlation coefficient. Then you want to see, okay, I need a cutoff. See, this correlation has to be greater than 0.75. I will keep it. Anything lower than that, I will set the connection between these 2 genes in the network to be 0, no connection. Okay, so this is like a hard cutoff. And they also give them some way you can transform, because there are negative correlation in there. How can you deal with negative values? So they plus times 0 point plus 0.5 and make everything positive. Okay, even you have a negative values, you push everything together, positive in there, then you set a cutoff. You can also use and transform it. For the older correlation coefficient, you take squared the power and then transform that a little bit and make larger correlation coefficient relatively larger compared to low correlation coefficients. So to stretch them out, so you can set up a cut off easily. And you can change this parameters. So I want to be more stringent, then you change the beta value a little bit, you get slightly different results. Anyway, so that you transform the fully connected correlation matrix into adjacent matrix by introducing a lot of zeros. Okay? 0 means those 2 genes are unlinked. Well, the highly correlated gene, they are linked. And then what they found is this naive network is very noisy. So they did a transformation. They transform every values in this network built from a correlation coefficient and using a way and then to compute the similarity between the topology property of each gene. If these two genes in this raw network have the similar topology, and then they are linked. So they through this process and get a so-called TOM similarity matrix. This is called topological overlap matrix. Okay, so from the co-expression network, adjacency matrix, and the transform and get this topological overlap matrix. And this matrix you can consider as a denoised version of the original one, okay? Because of the connectivity between genes are not by correlation anymore, it's by their similarity in terms of their topology values. And so this is a description of what is a topological overlap measurement. The idea was not invented by this package's author, but it's by another group. I think this is a Bababasia group, published in 2002. And once you have network, you translate this adjacency matrix using this equation, become a topological similarity matrix. And then you treat this as your original matrix. Okay, so this is one more step, translation, transform. And that's the equation to compute topological similarity. Again, this is two nodes, I and J in the network. So how can you compute this similarity between them? If they're same itself, you don't compute it, it's one, okay? If you have I and J, it's are different nodes, then you found the nearest neighbor, the connected neighbor of I and the connected neighbor of J, and see how many of them are overlapped. So if you have two nodes from one end and another, the direct connection, there's no overlap, so then you have 0 here. Well, if you have two nodes close to each other, they shared a lot of neighbors, then the count of neighbors in here. Okay, so Aij is whether they're linked or not, and then divided by the small minimum number of this. See who have a list neighbors, and the +1 minus Aij. So the idea was from here, okay, and they borrow this idea and to compute a new values between I and J. So at the beginning, you have a co-expression network. Co-expression network is adjacency matrix, see whether I and J, these two genes are linked, but then you replace this matrix with I and J recompute using this T, okay? So I'm going to give you an example, see how can you compute network topology similarity between 1 and 2, these two nodes. So this example is you have node 1 here, and node 2 here. They are not directly linked because through 1. Okay, so this is just the example. We are going to compute this T value
topology similarity between 1 and 2. Of course, in your application, you compute every pairs, okay? You compute 1, 2, 1, 3, 1, 4. To reconstruct this matrix. But I'm going to just give you one example of the compute this. So according to the equation, the value here is so the neighbors of I and join with neighbor number of A, okay? And plus A I3, then this lemma. Remember that part 1 minus. Remember that.
Anyway. +1 minus A.
First, AIJ in our case, not linked, so AIJ is 0. Now here is zeros. 1 minus 0. AIG not linked. Well, this value is how many neighbors they share. So A has neighbors 1, 2, 3. 2 has neighbors 1, 2, 3, but they only share the 2. So this share this here. And this value is 2. Share 2. And then the minimum, the neighbors. So 1 has 3 neighbors. 2 has 3 neighbors. So they are the same. So the value is still the 3. Let me see. I think that's 4. Yeah. So this value is 4. 1 has 4 neighbors. 2 has 4 neighbors. So you just follow the definition and replace those values. And then you get 2/5. So that's new value. Let's look on the floor. Okay. The same way, this, for example, you can replace this value. Okay.
So this is just small examples to illustrate how you compute topological overlap measurements. By the way, this is topological overlap, only consider one-step neighbors.
Okay, you can do for 2-step neighbors, the same definition. You use that 2-step neighbors, and for the 1, you consider the divided neighbors, also another level neighbors if they that would become. So this one is also the same. There's some overlap between those 2-step neighbors. So you can expand this definition from just 1-step neighbors to 2-step neighbors or more. You can imagine, and this method just starts smooth out the links between 1 and 2 with the share a lot of the neighborhood. Okay. Although they are not divided, but if you come to this, it's going to have so some kind of connections.
But as I said, so this technique is just one of the way to smooth out the signal. So they called this generalized topological overlap matrix at 0 times 0 level means it's original adjacency matrix. And then if you do 1-step smoothing, then you get something like this. And then if you do 2-step smoothing, you get topology like this. When you do 3 steps smoothing, you get something like this. Okay, you start to show those modules, right? Start to look at all the genes share the same kind of topological neighborhood relationship, you start to look at, okay, those are the neighbors. So they can recover this from this very sparse network to start to recover some of this highly connected modules in the network. So once they have this topological matrix, similarity matrix, they start to do this classification using a tree building approach. So if you use adjacency matrix, you get this unclear cutoff. See, I want to cut from here, this very narrow, very short distance allow you to set a cutoff. Well, if you do this with a topological similarity matrix, then you can build a tree like that to give you very clear position where you want to cut into multiple different clusters. Well, here you just don't know where to cut. So this is because you have a denoised network. So from here, you can start to detect these modules by using this clustering method. So all the genes in here is one cluster, all the genes here, another cluster. Okay. So once you have a cluster, they start to compute functional, okay, so whether genes in there enriched for some categories of the terminology in the function, and then to finish the annotation. They can also link with external information, so whether those group of gene show some level of the increase of it or decrease expression in particular categories of your samples. Okay, so that's basically WGCLA. So next time you see this, don't feel surprised. They have been cited thousands of times. Okay. All right, let's take a 5 minutes break.
Like, what's it specifically from the data sets that we collected, or this the so it's from the other end, or you put them in?
Yes.
Of a particular condition and something before like really collected. Is that what's for?
Yes.
Yes, it does that, or like you have to do the I think it's really see the labels on them for the cluster version. Oh, you got the cluster. Here.
Here.
Yeah, the one. Yeah, that's right. I'll tell you that. I get the ring to it. No, I didn't. I ported like real ported. Then it, it's one ring.
Okay, so we are going to continue with so-called sub-network identification or module detection, or different ways to call them, or community detection. The idea is in the large network, you can start to consider some of the nodes are highly connected to each other. They form a local module, okay? So then once you have, I don't know if you can see in this illustration, we have 3 modules. And then once you have those definition is given, you can start to say, okay, who is the local module hub? Okay, they're in the center of this, not the whole global network, but in the center of each individual hub module. And then we can start to detect those intermodule hubs. So some of these nodes looks degree may not really high, but they are in the critical position, which in charge of this communication information flow from one or multiple different modules. So those are called the intermodule hub. So once we have this, we can describe some of the function of those hubs. In single cell data, we also create cell-cell network by cell similarity using KNN network. And then you can detect the modules from those cell networks. Those modules are cell clusters. Okay. So the most commonly used community detection algorithm is called the Louvain modularity detection. So this author is from University Louvain in France, and they just call that. And also there are many other different ways. And this method, in theory, should be computational intensive. So you don't know how many communities in there. You don't know how to assign the membership for all the nodes in which community. So this involves iteration procedures to optimize the selection of membership. You want to select all the nodes highly connected to one community where others belong to another community. So how can you quantify this separation? Okay, so this algorithm starts by from assign each node into their own community. So if you have 1,000 nodes, then you start by say, okay, I have 1,000 communities. Each node belongs to their own community. Then they start to compute so-called modularity score for the whole network. So here there's a data function. So if you have I and j belongs to two different community, you have a data value to be 0, or otherwise they belong to same community, you have 1. So depends on membership assignment, whether they're yes or no, you can have this. And then you go through all pairs in the whole network. You start to compute a value of this. Then you start to change the membership assignment and through an iteration procedure a little bit, and then you improve this modularity values until you cannot improve it anymore. Then you stop. At that time, you have a community assignment. So this is a very time-consuming procedure, and I believe there over the years people have been trying to use different ways to accelerate this, and now it's widely used community detection algorithm, and there are many different language implementations make it fast enough to be practically useful. Okay, so as I said, we can have the community detection, which once you have that, there are a lot of other analysis you can start to be reviewed. And in traditional comparison between condition 1 and 2, you have only considered one network. Okay, there's no modules. Then you can see, okay, some of these links disappeared in another sample. So this package is for this purpose. Well, once you have community detection, then you can start to look at those changes. For example, there's a community hub in condition control condition, but this hub is disappeared. Okay, there's no link. So you know there's something wrong with this sample compared to original. If you don't have a community detection algorithm, you don't found those kind of changes. But that's just one example. There should have a lot of different analysis we can do. All right, let's jump into single cell world. Okay, we're talking about the single cell gene regulatory networks, sometimes called the gene co-expression or gene expression network, because the regulation network is a strong claim that because you know the link between genes, their regulatory relationship, one gene active have to influence another gene's expression, but sometimes they are just co-expressed. They all regulated by the third-party genes or maybe conditions. maybe under different temperature, those genes are upregulated in among cells. So you still build those network. So in that case, we call it gene expression network. Okay. Gene regulatory network. Yeah. And so we are going to use a single cell gene expression matrix to create network. All right. So you see this a lot because we have this technology, we can get this expression matrix. Now, this is real data. Okay. Now we can, for example, to look at two genes. Okay. Then we can select two genes, start to look at their correlation in expression. For example, this is a magic imputed values. Otherwise, you don't see this beautiful correlation between these two genes. This is from lymphoblastoid cell line. We sequenced ourselves. So there's two genes, okay, REL, AICBA, and change this from REL. So this is a very clear relationship. They positively correlated. Probably one gene regulated the expression of another. Well, in this case, it's not so clear as whether there's clear regulation relationship, because sometimes you see the positive regulation, sometimes you see the negative correlation. Each point here is a gene, right? It's a cell. Okay. But what we found from this data is if you look at the third gene expression, okay, so this third gene, KRDM1, the expression level is indicated by this color bar. So all the cells in this region are highly expressed this gene. Looks like when cells have high expression of this gene, those two genes will positive correlation. Well, when this gene expression level is low among those cells, these two genes looks like negative correlation. So we can review this nonlinear relationship by putting three players into this like a three-body problem. The reason we can see this relationship because we have enough points. If we only have 20 or 100, you don't see this. Okay, so that's the power for single cell data. You start to review very detailed multi-variable relationship, oftentimes nonlinear. So once we collect enough of those 3 genes, 4 genes, even higher, and the pairwise relationship, we probably can reconstruct a real regulatory network by looking at the relationship, a pairwise relationship between genes. On your left is a real CABA-B regulatory network. There's people working on the animal or work on the cell line for many years. We start to build up these connections between genes. So this part is several decades of the efforts in order to figure out who regulate who and who supplies who. Okay. However, in single cell world, we can use just one single sample. So this data is collected from just cultured lymphoblastoid cell line. So we have all the genes expressions, so you can put the pairwise correlation relationship between all pairs of genes. The one I just showed you is this corner here. They largely positive correlated. Okay, so from IRF4 to I see the positive regulated. So that's this link. Okay, so what I found is these two genes, the positive regulate to each other, and the correlation globally this positive, so I put this right square indicate the correlation we see from the single cell data is consistent with this concept map, okay, the build network. And most of them, the positive, they all consistent. Some of this not. So, for example, this gene to RELA2, this RPDM1. The traditional model believe this positive regulate this, but what we see in the single cell data is negative. So that's why I put a green box here. But overall, you can see majority of this. You can get a consistent pattern from the previous database knowledge. So if you don't have this, you'll still be able to construct majority of this parts by just looking at those genes relationship, their potential. So what that means is possible, maybe just use one single cell data sample to largely reconstruct or recapitulate by a lot of regulatory relationship from the from unknown. You don't need any prior knowledge. So this is from a review paper to compare bulk RNA-seq. That you have a cell sample. If you go bulk RNA-seq, you get all the genes expression, you build the network, but this is not a cell type specific. Well, on your right, you have cell types, and you build a network for every network single cell type. So some link was from the green cells, some link from purple cells, some from orange cells. Well, in traditional way, you just put them together, you get a global network. Well, here you see, okay, these two genes were linked in cell type 1, but not in cell type 2. So you have this power to be cell type specific. So that's the advantage of using single cell data to construct networks. Number one is the sheer number of cells gave you a large sample. So thousands of cells definitely is one of the most powerful data set we can have in order to build a network. Okay. Also, single cell itself, every cell is different from another. So inherently and or naturally, they start to show this variability among cells. So to start to show the variability, you have to have this variability in order to capture the covariance, co-change, co-expression between genes. Otherwise, you don't have that signal. So single cell data just automatically provide that variability. That means, so even you just collect the cells in one stage, but the cells itself in different in the different states. We can also collect, for example, at multiple time points. In that case, we really follow the dynamic change from one point to another, especially if there are development associated, or you can sample from different conditions, but those conditions are continuous variable. So this is anyway. So as you can imagine, this is really give a lot of advantages to use this. In this review paper, they also say, okay, we can do clustering first. Then you have cell type A, B, C. You do cell type specific construction of networks. You can start by trajectory analysis. So you use pseudo time to align cells and give them different pseudo time points, and then you can follow the change of the dynamics steps. So you have a network at time point 1, 10.2, 10.3. So you follow the change of this those network regulation change events. And then you list some of the genes you can use, methods you can use to build networks. So this is what I mentioned. If you build the trajectory for this to give you the time series information to start to build a temporal dynamic aided network, the dynamic network. So there are some commonly used method, and our cell, we did a single cell 24. This package, with this package, we use so-called PCMAT. It's called principal component regression. So we have some internal tests. Our method is really in terms of the balance in the accuracy of the construction and also computational efficiency. A very balanced method. So we should use PCMAT, principal components regression all the time. There hasn't been involved in a lot of those benchmark testing study because we published this method relatively late, and some others already published other methods. So they get involved in this kind of a benchmarking algorithm to test whether which method can construct network more accurate. For the ROC curve, okay, to help you to understand the accuracy and the specificity of the construction, and also other methods. We also consider the time, running time. So this study is called the B-line evaluation. So they call them the B-line study. So this framework compare the performance for many different algorithms for single cell gene regulatory network construction. Okay, so again, they gave this the evaluation results. The problem for this is what they used as a ground truth to evaluate accuracy is their input network is too small. So for example, this is a large network they have. I don't know how many genes in there, but could be 30 genes or 25 genes only. Then they even use even smaller test network to evaluate those methods. But in real application, we try to build network for thousands of genes by thousands of genes. Okay, some of those, I think a lot of those algorithms just not give you any results in the reasonable time frame. It may take a week to compute a 2000 gene by 2000 genes network. So I don't believe those construction evaluation, but just keep that as a reference. Some of this may help you to write a manuscript and say, okay, I want to use this method because it's evaluated in this B-line study, and it's one of the best. Those are options, different methods they tested. One of the method is called the Genie3. To me, it's a very strange implementation. And number one, it's used random forest algorithm to construct gene regulatory network. Random forest method is one of the classification algorithm. Okay. So once you build, you train your random network to classify, and the input you give the input of which class it belong to. But they use this framework to do regulatory network construction for the gene network, and for small data set perform very well. And in some of this competition, they always give the best results. So we need to know a little bit how this algorithm is working. By the way, it's slow, so you have to be patient to build large networks. Okay. So that's the algorithm. They will take out one genes and use all the other genes as the input variable to predict the gene you just take out is the expression level, and which gene is more important as a variable to predict this gene's expression, then that gene will be strongly linked with this gene. With the second contributing variable, the second gene will be also linked, but with a smaller weight. Okay. So you repeat these procedures by take out a gene and compute all other genes' contribution to this, and you take another gene, contribute other genes to it, and you repeat this procedure until you loop through all the genes and start to build a network. Okay, so the principle behind that is we can use tree as an algorithm to do classification and regression. So the whole idea is using decision tree algorithm. This authors have a book about that. So this is called classification and regression trees, or CART, for classification and regression purpose. Let's see how this model is can be used to do the classification. So in this binary tree, okay, you start to answer the question, and you have variables, for example, the height and weight with two variables. You want to predict the sample, is there male or female? So you have the height variable that you start to select, so 180 centimeter as a cutoff. Yes or no, you separate your samples. And then you use weight, 80 kilograms. Yes or no, you separate the samples. So you have a breakdown using multiple cutoffs, and then finish your classification. So in this model, there are two cutoffs: is the cutoff for the height, the cutoff for the weight. So once you establish this tree structure and also these two parameters, you finish the training of this binary tree model. Okay, start to have new data comes in, and if you have a description of height and weight for this new sample, you put in this tree, and the tree will give you this male, female answer. Is that simple?
Yeah.
Okay, so now we expand this to multiple variables. In this case, only height and weight. You can have another many different. To represent this, you can see, okay, I can use variable x1, so there's one variable weight, variable x2 height, and then the y is category. In this case, it's multi-category problem, so it's more generalized model. Okay, you just imagine you have two variables that you use first variable to separate samples into two categories, and the second sample them until you build this different cutoffs and the different categories. You finish training, and you can use this whole structure as your classifiers. Once your data comes in, you know which position you will put them and which category it belongs to. The same idea for the regression. So now you only classify them into 1, 2, 3, those are bins. But if you assign bins into values, you do a regression. So once you have many clusters, the problem become regression. and just make approximate this continuous change. Okay, you just put a lot of bins in there. So this model just learn first from you select input variables and split points your samples based on these cutoffs of the variables, and they use the greedy algorithm to learn to train the model and then stopped and then build the tree. Another way to illustrate this, but in this case, we are doing regression. So you have the price, okay? Not only the bin, which bin there, but also the real values for those bins. And this same framework is being used as a regression tool, okay? So that's a very brief introduction of the algorithm itself. The whole idea is like that. But for the real implementation, I believe there are some way to accelerate how you are doing the search for the cutoff or the tree structure, where you want to set up a region point and on which variable first. Maybe they have some other customized way to do this. But the idea is you can use a tree to classify the samples. You can use a tree to do the regression. Okay. So Genie3 just borrow this tree regression tool to do this. As I said, if you have an expression data matrix, you have your cells, you have genes. Now I will take out one gene, okay, use all the rest of the genes as input variable to predict this particular gene's expression. So this is what I learned LS. What's LS for? So LS maybe is a learning space, something like that. You have a learning space. The problem one is to use all other genes to solve gene one's expression to do the prediction. You build a predictive tree, and then you move to another gene 2, and use all the other genes to predict gene expression 2, gene number 2's expression, and you build a lot of tree. And then at the end, you loop through all the genes. You have a P genes, and you repeat these procedures for P times. Okay, use all non-P genes to predict P genes expression. Then they start to assemble those trees, and then to give the power for prediction for which gene can predict the gene 1, which gene can predict the gene 2's expression the most, until end we have this rank for the genes, then start make links. Okay, so this expression, this tree construction and network construction method has been incorporated into a package called the Scenic. So Scenic just take this expression matrix and using Gene3 to build a network first. Then they search for this transcription factor and the target genes relationship. Because once you build a network, this network is built on the co-expression. They are not a real regulation relationship. So if you have a gene A and a gene B, they co-express, they linked, but they are both controlled by gene C. So they look like linked, but there are no direct regulation relationship between them. So this package just use database to say, I only keep those links between transcription factor and their target relationship. I ignore all other links. Okay. So once they have this, you can start to see, okay, as a gene's transcription factor activity site. So how many of this transcription factor and their targets as a multiple links contribution and to reflect your cell activity states. So we use another steps to find so which transcription factor is active in this particular cell type. Okay, as long as you, for example, this transcript factor 1, and there its targets at the whole site, and they assign a score for the cells. So those cells have a really high score in terms of this particular transcript factor, and they will say, okay, this transcript factor is active in this cell. If you look at TF2 and this links maybe not so strong, which is TF2 is not active in the cell. So they can have this type. They gave you so-called regular activity estimation for the cell. It's very useful, right? So that's some applications for them to see, okay, I have some pathway, some factors, positive accurate activity, and what are their binding sites as well. Okay, they're supported by all the database. And they start to see, okay, they can detect those changes. Any questions? No. On the Cynic, I found it very hard to use. Shren, you have experience to using this Cynic? How easy to set up the pipeline? Oh, Shren, you spend 2 months to set up this pipelines. That's not good. And I think the problem is you just rely on the annotation, the database. You cannot find any new regulation relationship. You have to be the transcript factor and their targets has been in the database. You start to compute the activity values for it. But if this relationship is not there, or some transcript factors we are working on is not in there, then you don't have this information. Okay.
They seem to have made it a little faster though, because they are the Python version now.
Okay, so it was developed in R and then they changed to Python version because
Called Cynic Plus.
yeah, makes it faster. But the Genius 3 by itself is very slow. So I don't think you can use this for solving the genes. It's difficult. But it's popular. Okay, so this is a different approach, so-called information theory based. So they compute so-called partial information decomposition, not rely on the correlation, but some kind of computation for so-called information theory, what that called? It found name. Anyway, so this is kind of called information measures. Okay. Yeah. So mutual information. So MI, this is the keyword I want to pass. They use mutual information and get the data from single cell and look at each pairs of genes, see whether they are linked through the mutual information. And they also consider unique, redundancy, and synergy. Anyway, at the end, they compute this information score, which combine the different types of association. Okay. What they found is you can recover nonlinear relationship. You don't have to have two genes always monotonically increase. So that in that case, you have a good regression. But sometimes you go up first and then go down. In that case, you don't have a good correlation, but the information theory, there's still two genes move together. Okay. Another is called S code. So in this case, you need to provide the time series. So you have to give the pseudo time for each cells, and they use some of these iterations and to help you to get this network. This is another package to claim you can use the causal in regulatory network. The idea is they use this time information. Okay, you have time somewhere. So you need to find the network, the multiple layers of network, and something happened in the previous steps should contribute to another step. So the information pass on through across the time, and then you have the inference of the causal relationship, and you cannot go backwards. So they take this advantage using leverage time series data to find the causal interaction. Okay. At the time when I cite this paper, it's still in the preprint, but now they publish it in the cell system. Okay. So I believe this is also very slow. I haven't tried recently. Okay, you need a time series data that be powerful enough to compute, take time to compute. Okay, so so this area is what we are actively working on. We try to consider multiple layers of network and see whether we can do multiple layer modulation, modularity detection, whether we can find network switching, but sometimes you have a module, next time you don't, so you switch those sub-modules. Whether we can use time varying network graph theory to learn something, and there are also multi-view negative, non-negative matrix factorization method. So we just treat those networks as multiple networks and see whether there are some method we can apply. We don't have any good solution. I don't know whether on the market there are other solutions, but some publications, many are not for a single cell itself, but they are for social studies. We try to see, okay, change of the modularity through the time, detect the dynamic community structure. And we try to borrow those methods. This is like a multi-layer modularity. Remember the Louvain algorithm, and this part, but they also build the other more terms to incorporate those. See whether we can model the change of the modular structure across the time. So this is like a switching. So you have one module here, and then after in the second window, this membership switch from red to blue. Okay, whether we can detect some of this. This time varying graph. Okay, if you don't have this time series data, you have the whole collapse network, but you don't know whether this link appeared or disappeared at which time point. But we try to model this. A lot of theory, graph theory, concept or formulas probably have already been proposed, but we are trying to adapt some of this in our single cell data analysis, but not yet got anything. So this last one is about this non-negative factorization, matrix factorization applications to this multi-layered network. And this particular method will be able to detect, for example, this module, which is here or here, we not changed, it's consistent across the time. So we try to find a conserved functional module in multiple matrix biological networks. Okay, so any other questions? So multi-layers networks, we don't have any good solution yet. Shrey is working on that, so sometime maybe later he can find some good solutions for kind of a modeling. Okay. So at the end, I'm going to, I think a lot of you already seen this, and who have seen this before? Have I mentioned this?
Yes.
Oh, mentioned, yes. I can't remember sometimes when I mentioned that which. But this one is for detect the difference between two networks. So you can build single cell gene regulatory network for condition 1 and 2. And for example, this gene, and it was strongly connected with the 3 other genes, 654, but the connection is still there, but it just become slightly stronger or weaker, depending on which one. So we try to detect the gene, which by comparing these two networks to find those genes have a significant change in terms of their topological or their regulatory relationship with other genes. So this is not a gene expression level change. So the expression level for G2 genes maybe not change at all. It's just this relationship with other genes changed. So the method we use is to build from two samples is a UMI matrix subsample cells, and for each subsample cells we use PC regression, principal component regression, to create these networks for each of the subsamples. So you can see if you subsample the original matrix by 10 times, you have 10 subsampled matrices, and here have another 10 matrices. And for each, you will build a network, but you at least build 20 N by N regulatory network. So the method you use has to be highly efficient in order to achieve this in a reasonable time. So once we have those multiple networks, we use tensor decomposition to learn the major components. So this is like 3D for each. So each of these tensor will have 3 major components. We will keep top components, and then to reconstruct by multiplicate those vectors and reconstruct a denoised version of this network. So you can take average, collapse them, but we found, okay, this tensor may be better, okay? And then you have another version from the sample 2. Then we have 2 denoised clean networks. We start to do manifold alignment to compare them. So after you align this 2 manifold, and then you found on this manifold of this the same gene appeared twice. One is from the sample 1, one from sample 2. If they are well aligned, that means their network position is very similar to each other. Well, on this common manifold, if this gene 2 positions very different, then that means they have changed a lot from one network to another. So we just found those genes have a great distance and collect them and to do this enrichment analysis. Those genes are called differentially regulated genes. So they are not DE genes. They change their relationship with other genes. Okay, so this is one of the example from the mouse data, and you don't see the difference between control and the treatment group. And those major genes, you don't see any DEs. They express in the same level across multiple samples, same neurons, but they use this 24-net comparison, you can get these modules of those genes. This is in the control. The regulation network looks like this. And this is in the treatment, become like that. So this module contains a lot of this morphin-related function. Okay, so we founded the module and include most of the DVG, differentially variability, differentially regulated genes. And this module contains the function of those genes, morphin-related. And also this, the treatment given to the mouse is morphin injection. And the data from the paper original authors, they didn't find any DE related to any function about the morphin addiction or neuron. So, but our method can find those exactly where and when what a relationship has changed. You compare these two modules, they're different. Okay, so this idea had been intended to build a virtual knockout. Have I mentioned virtual knockout? Not yet. Through the course, virtual knockout. So we can simulate a gene once you knock it out from the neural network. What other genes will be changed? Okay, yeah. So what we want to do is to try to replace this animal knockout experiment, because you knock out an animal, then you compare, for example, single cell data, the same cell type between one white type and the knockout cells. You get those genes which changed, and you look at the gene's function to see, okay, what is the function of this knockout gene by looking at this DE genes. But we don't have to do that because we can simulate knockout this gene, what other genes will change in the network to give you the information about this function of this knockout genes. Okay. So this is the idea, and this is exactly the same pipeline for compare the networks. But in this case, we trade this as white type, and this we only use one white type. Okay, this is white type sample. You can get expression matrix, but here we don't have the knockout. We just copy the white type, put in the as a knockout. Now, if you run this pipeline, you still get some genes, but those genes, it's just random genes because the input and output are the same two samples. But what we did is we found the gene that we want to study, the knockout that gene by set all the expression value of that gene in the matrix to be 0. Okay, so you get the same matrix. You copy the matrix and set the gene you want to knock out expression to be 0, all zeros cross, then you still run this pipeline. You still get the answer. And this answer is a comparison between two networks, one with that gene, one without. And most of the time, the gene you knock out will be on the top, the most different ones. And the second and third will be the gene, or the top 20 other genes will be the gene that will be greatly impacted by knocking out the top genes. Okay. So we can simulate this by looking at our simulated results with real knockout animal. They have a wild type and knockout real data to compare to get the gene function for that target gene. And we can do same thing in computer without using knockout animal information, just use wild type and still get the same results. Then we say, okay, we don't have to knock out animals. So the whole idea is you build a accurate, well-defined network. And once you have any impact on this network, you know where it is and how strong the impact is. So the last one is so-called interaction. So we do have this two cell types and constructed neural single cell regulatory network, and also whether they have communications through ligand receptor pairs, and then same kind of a manifold alignment to found ligand receptor gene expression and to find those interactions. Okay, so that's all for today. And at the end, I will also mention the progress made in my own lab. We are still working on the next one. It's called a single cell time. It's a time series data network modeling. Shred is working on it. Okay. Oh, thank you. And I will see you on Thursday. We will look at some network construction algorithms and practice the use. Questions? Okay, I have video recorded, and in the beginning, I introduced how this student presentation will be conducted. That will be next Thursday. So if anyone missed that information, you can review this video.
We have 15 minutes.
Yeah, 10 minutes and 2 minutes for questions.